Hello, welcome to our YouTube channel. We are discussing about the spermatogenesis, and till now we have studied in the previous lecture that um, the progenital germ cell reached to the genital ridge, and after it reaches to the genital ridge, there was two fate over here in the genital ridge. First was whether the cell will undergo into mitosis or meiosis. We have already seen that the progenital cell under the influence of RA, retinoic acid, undergoes the meiosis, but in case of male, in case of male, this retinoic acid, yani RA, was being degraded with the help of cytochrome 26, A1, B1 and C1. So after its degradation, the fate of meiosis could not occur, and those type of the cell will turn to be a male fate. Okay. So up to now we are clear over here that the male fat will undergo the subsequent mitosis. But till now, up to now, whenever the progenital cell reaches to the genital ridge, it is inserted with the sex code. It is inserted with the sex codes. Okay. And these sex codes this sex code will hollow out. This sex code okay, we were looking, this is what the genital ridge. In the previous lecture we have seen genital ridge. This one is the genital ridge, this one is the genital ridge, and these are the progenital germ cells. These are the progenital germ cells. What happens? It is get entangled into the cord cells. Okay, and these cord sex cells, okay, this cord sex cell will during the course of development will hollow out. This will hollow out to turn into the seminiferous tubule. This will turn out to be a seminiferous tubule, and this genital ridge, the individual genital ridge, okay, will turns to be this this one will turn to be the packet, and the cell after subsequent division will turn into the testis. Okay, so let us understand, let us start our today's lecture with the testis here. Okay, so here we can understand that, so this is a mature testis, over here. So this is you can say this is a vas deferens, this is epidermis, okay, and this is the testis. This is a testis, and whatever we are seeing here in the red color, it is a convoluted pipe is there, or convoluted pipe is there, and this is called as the seminiferous tubule. Just pick out the one end of the seminiferous tubule. After getting out the seminiferous tubule, one of the seminiferous tubule, just stretch it. After stretching, we will see this like structure, a hollow tube like structure is there, and if you are taking the cross section of this hollow tube like structure, we will get just this is this is exactly what it is. Okay, so this is hollow like structure. Okay, this is a hollow like structure, and after getting the cross section of this part, this very part, we will get this structure something like this. This is the internal structure. Okay, so this is the internal structure. I have taken a small part of it. So, okay, so like this, suppose if you are taking the structure over here, so we will see the structure will come like comes to be like that. Okay, let me draw here. The structure will come to be like that. Suppose this is what we are taking, this is what we are taking, and this structure will seems to be like this. Okay, this structure will seems to be like this one. ये structure जो है ऐसा कुछ दिखेगा. Okay, and there is lots of various type of the cells which are embedded toward the basal lamina of the seminiferous tubule. We will discuss after taking the zoom of this picture, and this is what we are looking here. This is the tail. Okay, so after taking the section of this area, okay, so after taking this is the hollow part, and these are surrounded with the various type of the type of the cell of various type of the stages of the sperm cell. Okay, so after taking the cross section of this area, we found something like this. I have just showing you the one part. Okay, as you can say that this part we have taken it into the consideration. Now let us understand what is this about. Okay, so this is the outer layer. This is you can say this is the outer layer. Okay, and here this is the outer layer, and in this layer we found the Leydig cells are there. Leydig, Leydig cells. Okay, and this is what we say the basal layer. You can say the basal layer. Okay, and this basal layer, okay, this basal layer, the epithelia, this is the epithelial basal layer of a seminiferous tubule. The epithelia of the basal layer, it differentiate to form what? The germs, to form the Sertoli cells. Okay, so this basal layer differentiate to form this Sertoli cells. Okay, and these are the progenital germ cells, PGCs, or you can say the stem cells. Okay, these are the progenital cells, or you can say the stem cells. Okay, and just for your information, whenever the progenital germ cells are inserted into the sex cord, it remains to be there in that state till puberty. After the puberty is attained, thus division and differentiation occurs. Okay, and after successive differentiation and division, okay, this stem cells is retained. This is undifferentiated stem cells. This is undifferentiated sperm stem cell. Okay, and this one will lead to its various fate. Okay, these sperm cells will have its various fate. Okay, so let us understand in a layman. Thereafter we will try to understand with the help of the various type of the factor associated with it. Now let us understand here. These progenital germ cells have the different fates. Progenital germ cells, or you can say the stem cells have the various fates. Okay, what are those various fates? Okay, this undifferentiated stem cells will undergo in two forms. Either they will differentiate or they will proliferate. Proliferate what? Proliferate क्यों करेगा? Proliferate इसलिए करेगा so that they could increase their number. And why they will differentiate? Okay, so differentiation will occur because to just to reveal out its successive fate of those stem cells. Okay, so we have the two type of the signaling processes which often occurs on the surface of the this stem cells. Okay, so what are those successive what are those signaling? That is the WNT signaling that we have already discussed previously, and another one is the BMP signaling. Okay, so BMP signaling let me wipe it out. Already we have got, so we have just let us first of all हम लोग यहाँ देख लेते हैं कि ये सेल क्या क्या है. सिंपल सी बात है. सो दिस आर द स्टेम सेल्स and upon the subsequent division this turn to be a different fit and this one to be a different fit. Okay, what are that different fits we will discuss one by one whenever we will reach to that part. Okay, first let us understand about this stem cells. Okay, the fate of this stem cells. Okay, the fate of the stem cell let us understand here. The stem cell will lead to this stem cell. Okay, either it will differentiate. Okay, it will this is the stem cell. Okay, either it will differentiate either it will differentiate or it will proliferate. Okay, either it will differentiate or proliferate. Differentiate means it will divide to its another fate, and proliferate means it will divide, or you can say it will renew the same cell. Okay, so we have the two type of the signaling. That is, the first one is BMP signaling. And this will lead to the differentiation of the stem cells, and the WNT signaling, will lead to the proliferation of the stem cells. Okay, means it will increase the number, WNTC will increase the number of cells. Which one cell? That is the stem cell. It will increase the number of the stem cell, and this BMP signaling will decide the further fate of stem cell. Okay, this will decide the further fate of the stem cell. Okay, now the question arises, which type of the BMP signaling is, or BMP protein is responsible for the differentiation of the stem cell? So that is called as the BMP8. BMP8B. This is the protein of the BMP. Okay, this is the subtype of the protein of the BMP, which is responsible for actual differentiation of this stem cells. Okay, now what will happen after it will undergo the differentiation? Okay, after the differentiation, this stem cell, okay, this stem cell in the form of the type A spermatogonia. Okay, after differentiation, it attains the type A spermatogonia, and this type A spermatogonia further divides. It has also different fates. This type of spermatogonia, type A spermatogonia, further divide to form the black fate and another the pale fate. Okay, so we will not go into that much details here. We can cover it in another lecture. Okay, so first of all, let us understand the type A, the spermatogonia will further divide. From here, we will discussing about the division of the sperm cells, or you can say the sperm stem cells. So this is the sperm stem cell, which is called as the type A spermatogonia, will further divide to its another fate. So what is that another fate? Let us understand here. So right now, up to this point, it is clear that BMP signaling and WNT signaling are the key factors for the differentiation and the proliferations of the stem cells. Now, let us move towards the another section. What will happen thereafter? Okay, so after the A spermatogonia, type A or spermatogonia will further divide to form its replica, that is A and A1. Okay, and after subsequent division, A1 will lead to the formation of A1 and A2. Okay, and thereafter, listen carefully, A2 to A3, and this A3 will lead to the formation of A4. So whatever we are studying here, so this division is occurring. That is what type of the division is there? That is the mitosis division. Okay, so this is the mitosis division we are going through. What will happen after? The cell A4, after the A4 division, it's further divide mitotically to form intermediate spermatogonia. It will further divide to form intermediate spermatogonia, and then to type B spermatogonia. Okay, so these are the stages of the division of the sperm stem cells. Okay, then after, what will happen after it divides to the type B spermatogonia? Another mitotic division occurs, and that will be the final mitotic division. Thereafter, the cell will undergo the first meiotic division. Okay, so what will happen after the type B spermatogonia? The type B spermatogonia will turns to be, the most important section is here, this will turn to be the primary spermatocyte. Okay, primary spermatocytes. So let us understand here, this one is the spermatogonia and this is the spermatocyte. Okay, so this is the spermatogonia and this is the spermatocyte. So there is a big question arises here. Okay, the big question arises here, that what are those factors which are associated with the formation of the spermatogonia to, or you can say what are the factors which is responsible for the transition of the spermatogonia to the spermatocytes. Okay, those factors are, first one is, okay, factors factors for transition to gonia to spermatocyte. The first one is SCF and the second one is G, D and F. SCF means stem cell factor and GDNF matlab the glial cell line derived neutrophic factors. Okay, so these are the two factors responsible for the transition of the spermatogonia to spermatocytes. Now, what does these two factors do for the transition into the spermatocytes? Okay, so what happens here, the type B spermatogonia is the key step, or you can say the division of the type B spermatogonia is the last step, or you can say is the last step of in the cell division of the sperm to undergo the mitotic division. Okay, to undergo the mitotic division. Okay, so here is the two fate. Okay, the increase in the concentration of the SCF, increase in the concentration of the SCF will lead to the differentiation of type B spermatogonia, this will lead to the differentiation of the type B spermatogonia to primary spermatocyte. But the most important thing is that there should be equal balance for the formation, or you can say the turnover of the spermatocyte and the spermatogonia. Suppose if there will be unequal balance between the formation of the spermatogonia and the differentiation of the spermatogonia to primary spermatocytes, then what will happen? At a time will come, then all those type B spermatogonia will be utilized. So in order to replenish, in order to fulfill the requirement of the concentration or the amount of type B spermatogonia, the nature has given an important clue for its maintenance. Nature has provided another protein that is called as the GDNF, and the most important thing is that SCF and the GDNF are the prognostic in nature. Okay, the concentration or the increased concentration of the SCF and GDNF will never occur. Okay, so whenever the increase in the concentration of the stem cell factor, this will lead to the differentiation, and whenever it will undergo differentiation, the concentration of the GDNF is low. Whenever the concentration of the ACF is high and the concentration of the GDNF is low, then at only that condition the differentiation will occur. Okay, only then the differentiation will occur. Then what will happen thereafter? The concentration of, if the concentration of GDNF is high, the concentration of the GDNF is high, then what will happen? In this condition, in that situation, the type B spermatogonia will keep on dividing or increase its number. It will keep on dividing and increase in number. Type B spermatogonia. Type B spermatogonia will keep on dividing or self-renewal of the type B spermatogonia under the increased concentration of the GDNF factor. Okay, so with this the galloping of these two protein will lead to the balance in between the utilization of type B spermatogonia for the formation of the primary spermatocytes and its maintenance of the amount of type B spermatogonia to manage the formation of the spermatocyte. Okay, so till now we have a clear-cut idea regarding this step. So what will be the fate of this primary spermatocyte? Okay, till now we have studied the division. Okay, so this was the last meiotic division, this was the last mitotic division. Okay, means from type B spermatogonia to the primary spermatocyte, last mitotic division. So this is the last mitotic division. Then what will happen thereafter? What will happen thereafter? So after what will happen, let us just wipe it out. इसको हम थोड़ा सा पोंछ लेते हैं. This primary spermatocyte will undergo the first meiotic division. This primary spermatocyte will undergo the first meiotic division. Okay, this will undergo the first meiotic division to form the secondary spermatocyte. To form the secondary spermatocyte, we can also denote this like this one and this one too, like 2 degree. Okay, this is the primary spermatocyte and this one is the secondary spermatocyte. So again the question arises, again the most important question arises, what are those factors again responsible for the transition of the cell from mitotic division to the meiotic division? Again, okay, mitotic to the meiotic division. Okay, so this is obviously the retinoic acid, retinoic. Okay, so again here is the Ra means retinoic acid. In the last lecture we have understood that the Ra, retinoic acid, is responsible for the fate to the mitotic division to the meiotic division. Okay, so here this again come as a dominant factor. So the retinoic acid is responsible for the transition of the primary spermatocyte to the secondary spermatocyte under the influence of the first meiotic division. Okay, अब now there is another discussion, matter of discussion कि at that time there was a degradation of the retinoic acid. What about this time? So there is no existence of the mesonephros right now here. The kidney has completely been formed, and we are talking about the steps here in the puberty cases. So the mesonephros has turned into different kidneys. So there is no any formation of the what? There is no any formation of cytochromes which are responsible for degrading. What are that cytochrome? The cytochrome P26B1, A1 or C1. These are the three factors, three cytochromes that are generally responsible for the degradation of the retinoic acid. So this type of the cytochromes are no more in existence. So that's why there is no any fear for the degradation of the retinoic acid over here. Okay, so there is no degradation of the retinoic acid. So in that case, the cell undergoes the secondary spermatocytes. Okay, the cell undergoes the secondary spermatocytes. And obviously due to the increased concentration, okay, so after the primary spermatocyte turns into the secondary spermatocyte, as the concentration of the spermatocyte increases, the concentration, the amount of the number of the secondary spermatocyte increases, the concentration of the retinoic acid also increases, and this leads to the another series of the division that is called as the second meiotic division, and this will lead to the formation of the spermatids. And this will lead to the formation of the spermatid. Till now we don't have any confusion over here. Okay, now this spermatids undergoes the second meiotic division to form the spermatids. Right now we don't have any confusion regarding all this flow chart. Okay, now let us wipe it out, and we will stop here till we draw a figure, another figure. So let us completely wipe it. इसको हम लोग पोंछ लेते हैं, साफ कर लेते हैं, and then we will revise, and then after we will proceed to another section after the differentiation or after the formation of the spermatids. So let us understand here. हाँ, चलिए टाइप ए spermatogonia, and this will ultimately lead to the formation of the A1. This will lead to the formation of the A1 or A1 type A1 spermatogonia. So there is a various steps regarding the transition of A spermatogonia to A1. Okay, there are the multiple steps as there. Multiple steps are there for the transition of type A spermatogonia to type A1 spermatogonia. Okay, now so this is the type A1 spermatogonia. Okay, now after its division, okay, so this is the A2. This is A2. Okay. Now this is the A3. This is A3. Okay. And thereafter we will come to understand this 2 will undergo and this one is the A4. This one is the A4. Okay. And just we will not take account of all those cells. Just let us take another one. This one. Okay. And this will form the intermediate spermatogonia. And this intermediate spermatogonia will lead to the formation of the type B spermatogonia. Type B spermatogonia, and then to the primary spermatocytes. Primary spermatocytes and this primary spermatocytes let us see, here is condensed form of the genetic material, and now this is compact one, and this is called a secondary. This is the secondary one. Now, and this secondary one will again divide first meiotic, and this will again divide second meiotic division to form, it will divide to form the spermatids. This is called spermatids. So these are the spermatids. And what will happen now at this time, this spermatids, the Golgi body. So this is the mitochondria, this is the Golgi body, the Golgi body. The Golgi body of the sperm cell, or you can say the Golgi body of the spermatids will accumulate towards the nucleus, 2/3 of the nucleus, this one, like this one. So it will be enveloped, the 2/3 part of the nucleus with the Golgi body. Okay, the two-third part of, just let us take one figure. Okay, the two-third part of so this is the nucleus. Okay, this is the nucleus. The two-third part of the nucleus, two-third part of nucleus is enveloped, covered by Golgi, and this will attain to form the acrosome. Okay, and this will form the acrosome, and this acrosome will have the different types of the lytic proteins which will further be responsible for the cortical reaction after the fertilization, or you can say during the fertilization. Okay, now what will happen thereafter? The Golgi body, the centrioles will leads to the formation of the, it will in next step it will again the centriole will okay, the centriole will accumulate toward the midpiece or the neck, and it will accumulate to form the midpiece of the sperm cell. So the centriole will aggregate towards the basal part of the nucleus. The orientation of the centriole will occur toward the basal part of the nucleus, and thereafter the mitochondria will wrap it. The mitochondria will wrap over it. Okay, and this will be wrapped by the mitochondria. This will be wrapped by the mitochondria. So right now we are studying over here the spermatids. Okay, after that the distal part of the centriole, or the you can say the tubule, the extension of the tubule of the centriole to the distal region will lead to the formation of the tail. So let us understand with another photograph. And this will lead to the formation of the tail tail of the sperm, and then after it will just come out from the envelope, or you can say the envelope of the you can say the residual bodies. Okay, so this could be the residual bodies, and this will come out, and the sperm, the fully formed sperm will come out, sperm, and this is the residual body. This is the residual body.